So we are going to look at this lecture. Today we're going to look at this lecture of evolution of Metazoa. The evolution of Metazoa part 2. So here we are going to describe that organism they moved from one cell to many cells, and how did they evolve. We in the last previous lectures we talked about uh, organisms evolving from protozoans, which we said that protozoans are the single cell organism. And we talked about protozoans, uh, the phylums of protozoa, which are the phylum Psychodina, the phylum Mastigophora, Coelophora, and Apicomplexa. We went into details on those phylums. I hope we explained a lot about it. So, evolution of Metazoa. Here we are going to look on things like what happened in the beginning. What happened in the beginning. So we are going to look at the theories. We are going to look at the theories. Like we said that there are theories which are there in evolution. We have the Syncytial theory and we also have the Colonial theory. So we are going to look at those theories because we at this stage we know that in the beginning there were single cells, and later today there are after millions of years, animals are composed of multiple cells. So we know that from today, like in this stage of this syllabus, we know. But the question is, how did they come into being? We know that how they came, they combine, they do everything, but there should be some scientific evidence behind. Therefore, we are going to learn about the theories. So how did cells make the transition from life as a solo cell to associated and cooperating with other cells, such that they work as a single cohesive unit? This is the question that we are going to ask ourselves and are going to answer it. Transition from one cell organism to multicellular was a huge step in the evolution of life on this planet. It occurred more than once within the domina eukarya, leading to 3 kingdoms of multicellular life: kingdom Plantae, kingdom Fungi, and kingdom Animalia. Today's plants, fungi, animals, and various types of algae are all descendants of separate transitions to multicellular life. Like every kingdom which was there, scientists believe that it was part of the transition. Like we started as colonial cells. After colonial cells, we moved to other cells, which are the algae. After algae, we moved to fungi. After fungi, we moved to Plantae. Plantae, we moved to Animalia. But these are, but we don't concentrate on that movement. Like we're just explaining that because of the transition of one from a unit cell, unicell to a multicell, it led to some other things like this. What possible processes could have led to evolution of multicellular animals from single protozoans? How organisms then became multicellular was a huge evolutionary step and is still under much debate. Several hypotheses have been proposed as how multicellularity could have evolved in animals. So we still have there are many hypotheses which are there, which are trying to explain. Yes, we are convinced that we are from single unicells. That's a fact. But how did we came into being? Retracai, are we made by ciliate? Are we made by flagellate? Are we made by amoeba? Are we made by apicomplex? That's now the debate comes from, a scientific debate comes from. Because some people look at that diagram, is a paramecium. Some people believe that you are from paramecium. Some people believe that you are from a flagellate. Some people believe that you are from amoeba. But where are we coming from? But now we are going to discuss the theories. So those theories which we are coming from, like I can side with a theory, but because I feel like I believe that theory, but you also can side with your own theory. But now I'll just explain the theories, but don't but I want you to listen to the basic theory, but I can also side with another theory. Like I get someone can just take a side because I believe in colonial theory. So when I explain it, I'll make it better, but don't try to make it better. When you're a student today, you need to write and do everything. When I don't side with any theory, but that's a question which demands you to compare the theories and which one is the best and explain why you say it's the best. So these are things that you can listen attentively. So the first theory is called the Syncytial theory. So the Syncytial theory was proposed by Hertz in 1953 and is supported by Hanson in 1958. You need to know these things, that it was supported by Hertz and supported supported by Hertz and also supported by Hansen and all those kinds of stuff. So those students who have paid for the material, we are the one who we are sending these notes to you. But those who didn't pay for the material, they just see them here. They don't say anything. Like even questions of biology, I sent to those who have paid. I hope the questions are good. We send the questions, everything. We send you everything to you. But others, like now, I'm not bothered to send to them the revision of animal. How many questions are there? 35 questions of animal. Or is it 50 or what? I'm not bothered. But for you, for those who have paid, remind me so that I can send this note so that you appreciate them. You appreciate them. Tata. You appreciate them. Look at them. But it was proposed by Hards, supported by Hands-on. You need to know it. I can't explain this. I can't, but you need to be reading down and you highlight it with some color so that you don't forget it. So according to this theory, this is the part we need to know about the Syncytial theory. The Syncytial theory, it assumes that metazoans arose from the group of multi-nucleated Syncytial or ciliate that assumed a benthic lifestyle. Benthic, we said these are organisms which are attached to other organisms deep down in the ocean. Sometimes they can be deep down the ocean, but sometimes they can be attached. It can be the word to use for attached. So Hards and Hanson, they are saying that, ciliates, all of you, because you've learned about ciliates. According multinucleated ciliates, we mean that these were ciliates which had multi-nucleus. It has many nucleus. So Hards, they're saying that they just believe that there was a multi-nucleated ciliated protista, and that multi-nucleated protista, I don't want to. It is a single cell. Is a paramecium. Is a single cell like a paramecium. You can zoom. Try to zoom the first picture. Try to zoom. Is a single cell like is a single protista at the top with the number one. You are zooming. You see that there are dots inside. Let's assume that those dots, these are the nucleus. Hards and Hansen, that's in that. That nucleus there, take one dot. Take one dot. So that all of those dots, the dot is just circle them and make a cell. Circle them. I don't know all this circle, you are that structure, you are dividing it into many something. Like it is like you compensate the cell. Mind you, that protista there is a single cell. And what's a more circular nucleus in dotting you are making many cells. So that's why I say hard and Heisen that believe in that a single cell it became multicellular because a cell divided into many cells. Like those nucleuses, they became many cells. It became many cells in those syncytial theory. So we say that the surface nuclei became partitioned off by accusation of cell membranes producing a cellular epidermis. What the partition? It means that paramecium, it became divided into many cells now. I told you to draw the circles on top of that. So it became divided into many cells. Surrounding the inner syncytial mass, similar to the archaeal flatworms. Until recently, it was thought that the inner cell mass of the archaeos was syncytial. Archaeos, these are assimilate. Assimilate, these are animals without a coelom. We talk about a coelom later. So this structure, why do they believe like that? They believe like that because they were looking at the archaeal flatworms. They're looking at the flatworms. So the structure of a flatworm sometimes is like that. The flatworms like it's a flat and it's like it has divided tissue, divided it like divisions inside of cells. So when the Hans and Hanson saw that, they believe that maybe you are from that animal. So they believe that the flatworms are the first animals. But according to evolution of animal, the flatworms are not the first animals. The anthelmintics. The anthelmintics are not the first animals. The first animals are the sponge. The sponge. So you can see that you can't say if because of the flatworms are the primitive animals, are the first animals, according to Syncytial theory, because we want to support a nucleus in it might divide the high. Whereas in the chain, number 5, like now you can't say you have born your mother. Is it possible to say you have born your mother? These guys are saying that. So it does not make sense because the animals which is in number 6 or 7 in the hierarchy of animals, like you can say it's the first one. So it means that this theory, it has some faults. It has some faults. So the theory says that the flatworm is the first animal. But let's look at this essential theory. The credits of the essential theory. Credits are something which we can say, but here the theory was making sense. The theory was making sense. So arguments that support the essential theory were similar between the modern ciliate and the actual flatworms in terms of, like, I think we know that the ciliates are the first animals. Yes, there are some ciliates which are living today, but I can say that they've also evolved, but they are living today. And there are some actual flatworms are living today. So when you compare the actual flatworms, like the flatworms that you know from agriculture, and we compare also the ciliate, there are some structures which are the same, like the bilateral symmetry. But bilateral symmetry means that you can cut between into two. Bi means two. Lateral means a cut, but a cut which is not medial. A cut which is not in the center, but it is a cut. Although that cut might not be in the center, but that cut, lateral means a cut. So bilateral symmetry means alike. After a cut, you are alike. So you are cutting. So when you cut a ciliate, that ciliate, and you also cut flatworms, those parts are going to be the same. Like flatworms, you can divide them into two, and the other side are the same like the other side. Like you human beings, like we can cut into two, the left and right side are the same, they're bilateral. So when Hugs and the Hansens, they saw those things, they said, why? We are from the ciliate. The anterior and posterior axis. Anterior means at the front. When I don't want to say front, I say anterior. Posterior means at the back. Post, at the back. So they have this anterior and posterior axis, back and forth. Back, you know that flatworms have the back and forth. If it is, they have back and forth. There's a side where you can say that no, this is the head side, and this is the back side. Back and forth. The size, that also in terms of the size. Mouth location, that one there's still a debate, but it can be one. But I guess those animals, those animals body ciliate, they're so small that that structure, that DNA sometimes can just change in a mouth. It can be at the top, can be down, but after some time they can go back to normal positions. Those animals. So surface ciliation, they all have ciliates. They all have, some of them they've ciliate, like some they've ciliate inside. Even some human beings they've ciliate inside. They said, yeah, you see, we have ciliate inside. It means we are from ciliate. Mind you, there are 4 types of protozoa, which we need to choose where we are coming from. Are we coming from ciliate, flagellate, amoeba, and apicomplex? But amoeba is totally out. It can't be coming from amoeba. That one is totally out. Yes, there are theories that we are coming from amoeba. There are some mad scientists that are there which are trying to explain, but you are not going to learn about them because they may destroy you or disturb you. So the surface ciliation, they said, so these are the points which they credit. You need to know these points. Or because bana ba lebile mabaka ba lebile ya bo size, bo mouth location, bo surface ciliation, bo bilateral symmetry. These are the things that they were looking at, and they said, yeah, I don't want to the flatworms so and jaga ciliate. Mind you, flatworms are metazoans. Flatworms, those are animals, metazoans. So they've multicellular. So they had to compare about that one, and they people believed them. We have the drawback, reasons why we disqualify the theory, reasons why these are the reasons why we can stand and say, but this theory does not make sense. This theory does not make sense. One, the theory suggests that aciluminate, triploblastic flatworms as the first metazoans, but fails to explain the origin of more primitive Cnidaria and Ctenophora. Aciluminate, you're going to look at these terms. You can just underline them, aciluminate and triploblastic. Aciluminate means you don't have a coelom. You don't have a coelom. So triploblastic, we are made of 3 layers. We are made of the epidermis, apo, meso, and which is the other one, which is the inside. There's another dermis which is inside. Actually, we talked about the developmental. We said that there are 3 layers formed. There are 3 layers formed after gastrulation. When a gastrula forms, there'll be some 3 layers, germ layers. We said there are germ layers formed. I hope you still remember. I hope you still remember. Don't forget these things. So the theory, it cannot explain. I was given an example that the Cnidaria and the Ctenophorus, that the more primitive, but the Ctenophorus are not going to learn about them most of the time, but they are like the Cnidarians, but you're not going to learn about them. Similar spongy, sporozoa, another sporozoa, porifera, spongy. What's the Cnidaria? What's the Ctenophora? After the Ctenophora, what's the name? Yeah, what's the flatworm? The number 4, no? Antihelminthics, they're antihelminthics. I forgot that name, but it's antihelminthics there. So, how can you explain number 4? And you can't explain where number 1, 2, 3 comes from. So, how can you explain something? Sorry, symbol number 4, you say it comes first. This is what I was explaining. That does not make sense. It means if you say, the Cnidaria and the Ctenophorus, you will see them. They have 2 layers. Some have 1, they 2. Like, yeah, they are so primitive. Like some sponges, they don't move. Sponges, they don't move at all. So there's some sponges that don't move. They're like plants. They're so primitive. They're so old, they're Asian. And they don't have even for you to have an acyluminate and to be tripoblastic, it means that you have developed, because there's some people, it means that you are multicellular. I told you that there's some animals which are operating at a level of tissues, like the Cnidarians and the sponges. That's the tissues. They're operating at a level of tissue, but the animals. But you take an animals which have organs, you say it's the first animal. What are you saying about those animals which don't have tissues? I mean, those animals which are still operating at a level of tissues. What are you saying about them? So this is a drawback. This is the drawback of the theory. Yes, the theory, they suggested a good theory, but how can you explain number 4 when there's still number 1, number 2, number 3, number 4? It cannot explain the radial symmetry of co-illiterates. There's a radical symmetry. A radical symmetry is a symmetry whereby you like. Have we ever seen like what's taking a circle, you divide into 2 into halves. Radial auto like something like a circle, a form of a circle, into many directions. I don't want to circle for you to make an equal halves. So something like radical. We're going to look at some radical symmetry. According to this theory, bilateral symmetry of acyluminates should be the primitive symmetry for the metazoans, and radial symmetry being a secondary derived. Flatworms are bilateral symmetry. They are bilateral symmetry. They are also acyluminates. So we believe that organisms and this is radial symmetric because they're just cells. So this theory suggests that you see the symmetry of human beings, example, is bilateral. It means that organism started as a bilateral land. They didn't change the loser symmetry against the whole. Is it true? No, because we are seeing organisms like you can't say that organisms which are bilateral are the first living. This theory suggests that when the first animals were so developed, so they grew down. Does not make sense at all. Does not make sense. So the other drawback of the Syncytial theory is that there is no evidence on cellularization were found during autogeny, origination of development of an organism of lower metazoans. Cellularization is a process whereby they believe that a nucleus just like the the cell membrane just appears, that just circles around the nucleus. There's no evidence. Small animals were taken. They don't even select. They don't have many nucleus nowadays. I think at first you guys drew the paramecium. How many nucleus did you see in the paramecium? Did you see many nucleuses? No. Seems like there's no something like cellularization, making of cell within an organism, where we believe that an organism it has many nucleuses, so it has many nuclei. So if it has many nuclei, the cell membrane will just develop. There's no evidence. It does not explain the origin of metazoan flagella and the general occurrence of flagellated sperms in metazoans. In nowadays, the metazoans, they have flagellate. Many cells have flagellate. You can't say that you are from a ciliate, because if you are from a ciliate, where does the sperm come from? If you are from a ciliate, where does this cells coming from? There's some cells called the sporocyte. The sporocyte are the cells inside a sponge. They are also have a flagellate. Where are you saying those cells are coming from? Let me tell you something. The mastigophora, it can change shapes. The mastigophora can change what? Shapes. But ciliate, they can't change shape. That's stationary. So if you say that organisms are coming from ciliate, you can't explain the presence of flagellate. You can't even explain the presence of phagocytes, because they are operating like amoeba. Where are they coming from? Phylogenetic analysis based on morphology and gene sequences show no relation with multinucleosides. When you take our phylogenetic analysis, you take our development, we do not show any relationship of multinucleated cells. Because if we would show a relationship of multinucleated cells, it means our zygote should have many multinucleated cells. Multinucleated cells. There is no a multinucleated cells in all in most of the animals, except the flatworms, that the ones we have. But you don't have a multinucleated cells. You guys, you drew cells in biotopol 1. Did you draw more than one nucleus inside the cell? You said it's an animal cell. No. So it means whatever those guys were saying, it was a dream. They were dreaming to be scientists. I'm sorry to say that. I'm not saying I'm against it. No, it's a theory. We have accepted it. And is it okay? It's good there. Let's move on to another theory. We have another theory called the colonial theory. The colonial theory, it was conceived by Witchley, Lakenster, Haeckel in 1874. Later modified by Metchnikoff, 1886, and recently reviewed by Heyman in 1914. So it has how many people? 1, 2, 3, 4, 5 people working on it. And those people, they've like a year between working, like 100 years. So a century. A century. They've a century. It means after a century, somebody comes and modify it, and it still makes sense. What does this theory say? The colonial or the flagellate theory postulates that animals evolved from flagellated protista that lived in colonies. Here, the colonial theory says that the flagellates came together, the volvox, the pandorina, they came together. When they came together, they formed the colonies. I mean, those volvox, these are colonies. Like the pandorina, the uderia, these are colonies. So they formed these colonies. So after they formed these colonies, we developed from that colony. So this is the classic theory of origin of metazoa. I wish something is classic, it means it is the most relevant. classic is the most relevant. When you say this is a classic something, is the most relevant in the era that you are living, in the time which you are living. Like it holds water. That's why you say is a classic theory of the origin of Metazoa. So the colonial theory, this is a theory which I was explaining, like when I said this bio 112, I was just explaining that we are from this, we came to colonies. This is the theory which you're explaining. So it's simple and easier because the theory came about through association of many one-cell flagellate individual forming a colonial organism. With the increase of the number of cells, they became more and more specialized in structure and function. Individuality in the cells was lost, and the whole colony itself became a single multicellular individual or a metazoa. So here what you are saying is that cells came together. After some times, they began to specialize on some characteristics because there were now many. So the cells which were outside, they now specialized in absorption. The cells which were inside, they now specialized in controlling. So at the end, they began to grow large, like the colonies became to grow large. They lost some characteristics. Now they became specialized. So when they're specialized, it means integration took place. Integration is whereby now you combine or you work in a system whereby you depend on one another. This integration, you begin to work in a system whereby I depend on you and you depend on me. Not because you are symbiotic. No, it's not about symbiotic. It's not like the bacteria which is in animals to process cellulose. No, like integrations whereby you become a one. You become one. So the system is you are integrated. The system becomes one. So, according to this hypothesis, with time, the corneal cells became more and more independent. Cells started to specialize to perform different tasks and became differentiated and could no longer survive on their own. Why? Because now they depend on other cells. At this point, they would no longer be a colony of cells, but a multicellular organism, a very simple animal. This is how we believe that the simplest multicellular organism came into being. This is how we believe that a simple multicellular organism, simplest multicellular organism came into being. So this theory has been widely accepted since what are the credits of this theory? Since it conforms with the effects of embryonic development. It's composed with the effects of embryonic development. Look at embryonic development. 2 cells, 3 cells, 8 cells, 6 to 32 cells. These cells, they specialize, they form a coelom, that change. Like this is part of the way we are growing is is the same as the way we came together, as our cells came together at the first. It's also supported by effects such as the flagellates are highly plastic group, which is capable for great changes. Now, you can't ask me where are the ciliates in our body coming from? It's because the flagellates can change into ciliates. They are plastic group. Like they can change into many forms, the flagellates. But ciliates, they can change into many forms. Nowadays, in our body, we have different kinds of cells. We have bone cells, we have other cells. They're not the same, those cells. We have muscle cells which are like they're linear. Why are they linear? It's because like the flagellates can stretch, change, form a box, form all kinds of things. So another thing, flagellates show a great tendency of forming compact colonies. And these are clearly resemble various stages of embryonic development of metazoa. Flagellates, when they form colonies, they form compact colonies. That's why when we grow up in embryonic development, our eight cells, our morula there is so compact. It's so compact. So it's just it is proving this colonial theory. That's a more fact in more books which you need to be discussing. And other fact is that the metazoan tell the sperm, the presence of a sperm in the metazoa. All metazoans have a sperm, and the sperm it also has a flagellate in all animals. But you can see that, yeah, wow. Because of that, we can say we are from the flagellate. And also, the flagellated body cells are also common in lower in lower metazoans like sponges and correlate rates. Sponges produces what you call flagellated larvae, also possesses flagellated cells. We have cells called the chinocytes. Chinocytes are the cells on a sponge. We are going to study about them later, the chinocytes, later on in the chinocytes. So this chinocyte, the presence of chinocyte, it means that you are from the flagellate. It just resembles that you are from the flagellate. And mind you, I will try to post the notes of sponges so that you guys need to be reading them. And because we said that there should be presentation, I will ask you to present about. I'll give you a task now to present on, and I'll be listening. So according to the colonial theory, Metazoan ancestors were like existing freshwater volvo seed phytoflagellate. So I got the volvo seed, like a volvo, like a colonial phytoflagellates. So phytoflagellates means these are plant-like, meaning that the plant-like flagellates or protozoans are the first. These are our ancestors. We are from the plant, then became animals. But these are plant-like organisms having a cell walls. They have a chlorophyll and they have a autotrophic nutrition. The theory does not explain how these plant-like characteristics were lost during the course of evolution of the Metazoan ancestors. Because the question that we need to ask ourselves is that if we were plant-like, if we are plant-like with chlorophyll, with all this, how did this characteristic get lost during the way? This theory, it can't explain how we lost that characteristics. The reason why it can't explain is that there are some organisms which are between the volvoseed phytoflagellate and also the zooflagellate, which they can't recover. Mean that we don't know those organisms, so they're not even there. Like we can't recover them back, so we can't explain that change. But like if you would recover this organism back, those one are between changes, maybe would explain. But hackers like Insta, Michnikov, they were so honest with us and they said, we can't explain now. We change from that. They're not like other people who lied. I'm sorry. They didn't lie. Syncytial is true. Probably metazoans arose from some groups of existing zooflagellates having colonial organism. So probably, this is now our view. It's not part of the colonial theory, it's our view. Like probably, maybe there arose from zooflagellates, but the colonial theory states that there arose from phytoflagellates, and is convinced about that. So the flagellate protozoans, they have different shapes. I want to show you that flagellates have different shapes. They can be linear, they can be tree-like, they can be spherical, they can be plate-like, they can be solid, they can be hollow. You can see that they exist in many cells. I mean, in many shapes, proving to us that all cells of the body, they're made from flagellate. If you bring a hollow cell, I say, that is from a flagellate. Like we have hollow cells like the xylem. Also those they evolve from flagellates. we as plants and animals, we share this, the ancestor, the velocity, the phytoflagellate. We share it. Of this, which type gave rise to the ancestral Metazoa as being a subject of great speculation among scientists? But Hella, it is a part of the sounds. They can, but the main one to say really is a problem. But Hella, you need to know that the flagellates, the phytoflagellates that are the one who led to everything. So animals, they moved from protozoan to colonial, then to metazoa. Protozoan, these are single cell organisms. Protozoans that are made of one cell. We say that the one cell is a whole organism. We say that the cell performs all function in protozoa. We say that there's jack of all trades. Jack of all trades, I explained it is whereby you do everything. You do everything. You respire, you reproduce, you digest, you excrete everything. So the size restriction imposed by physiological processes. Protozoans, there's some certain limit to which they can't grow. So for them to grow, they need to come together. So they made what we call colonial. So colonial cells, we say that they're made of more than one cell. They are formerly independent cells. They had to learn from how to be civilized to avoid conflict. Cooperation of cells for mutual benefit. No cellular integration. Cells still maintain their individuality. So colonial organisms say that no, they're just helping each other, but they don't share anything. Even if you can separate them, they will live by their own. But metazoans are multicellular organisms. So they're made of specialized cells. And they've integrated. They are integrated. So metazoans that are integrated. They are integrated. So you read that, and you also, I've explained them before. I know now I'm boring people, but this is how, you need to, as a tutor, you need to be looking at what is key, so that when you reach to it, you know it better always. So we move from single cell organism, protozoa, colonial, and metazoa. So the advantage of single cell organism, the advantage of colonial organism, and of metazoa. These are the things that I want to read, because I want you to come and present about them. And we look at the disadvantage also. What are the disadvantage of single cell organism, colonial organism, and metazoa? So Uneile, tell them in the group then WhatsApp that our assignment is to look at the advantages of single cell, colonial, and metazoa, and also the disadvantages of single cell, colonial, and metazoa. And I'll have a lesson and you'll be presenting about it. So I'll just pick your name randomly and say, Uneile, present about disadvantage of colonial organism. You need to be in a position to discuss about them. Those who don't have network, I'll say a way how they will discuss it, and we need to understand. So tell them that the video, I've recorded the video, they may just watch. So it is how many minutes? I'm not sure it's how many minutes. So 48 minutes. So this is the end of our lesson today. And for Cam, you need to tell them that I'll try in the morning to record something like a 3-hour or a 2-hour, maybe a 2-hour lesson. And I'll go to where there's much internet, and I'll upload it in YouTube, so they should watch it in YouTube. Because having it as a lesson going to disadvantage a lot of people. But I'll make also how to draw alkanes. I'll make a special video in a form of a lesson in Teams, and how to calculate Coulomb's law, to do questions of Coulomb's law. I hope now you are watching videos and you know how to calculate. And that there's Coulomb's law in 2 dimensions. Calculate it. So we also do other things. For mathematics, tell them that they should read trigonometry. They need to read trigonometry. Thank you.